Hi, everyone. I'm very excited to be here in Boston today to talk to you about my research work, which is about SCALPEL, which is a Nextflow pipeline for quantifying isoform at single-cell resolution. I used to think that my implementation of SCALPEL was awesome, but after doing the 2 days hackathon, I really think that there is a highway for improvement and integration of all the new features that have been released by Sequoia. So before going to the computational side, let's dive first into the biological problematic, which is about the regulation of the gene expression. So going into the cells, following the transcription of the DNA, we have the generation of an immature mRNA that we call pre-mRNA, and then we have regulatory process which occur on those immature mRNA to generate mature isoform. If we dive into those regulatory processes, we have differential processes such as the capping or the alternative removal of intronic region within those immature mRNA. And particularly, we have one specific regulatory process, which is the alternative polyadenylation, which cleaves the immature mRNA at distinct site that we call polyadenylation site and generate mature final mRNA, which varying in the terminal region. Why we want to focus on the quantification of isoform at single-cell resolution is because most of the available data set for performing single-cell studies as 3' tag single-cell data set. In those data sets, we capture the mRNA content in the cells. We do a library preparation. We map on the genome. And thanks to the incorporation of the cell barcode tag and a UMI barcode tag within the reads, we are able to map those reads on the genome and get a quantification of the isoform of the gene at cell resolution. So here the challenge is to use the same data set and estimate which can be the isoform expression still at cell resolution. So we need to use the mapping of the read at the genomic space and then estimate what can be the isoform expression. And thanks to the nature of the 3' tag single-cell data set, where we have a bias of the signal towards the end of the transcript, then we are able to access the isoform diversity, which reflect the alternative polyadenylation regulation. And why this is important? Because the different mRNA isoform which we generate following the alternative polyadenylation regulation are targeted by different factors, such as RBP proteins, mRNA, which have an impact on the fate of those mRNA. So depending on the profile of isoform we have, we can have a different stability of the mRNA or translation, or even the localization of the protein. And more and more studies have been published recently, which point the fact that alternative polyadenylation is a very used process into the brain, and also it has been associated to certain disease. So then we can switch to the computational side. What I've been doing in my research work is to develop a tool which is called SCALPEL, which can use a single-cell data set, and then by doing a different set of operation, estimate the isoform expression at cell resolution. So I developed SCALPEL into 3 main workflows. So in the first workflow, we take the annotation, the genomic annotation file, and we apply a set of operations on it. Then in the second workflow, we take as input the read information, the BAM file, where we apply a different type of operation. Especially, we can see that we take an internal priming file, which are locations in the genome where we think that the mapping of the read are not relevant, and then we want to discard. And then in the last part of the pipeline, we perform a set of analyses, such as EM algorithm and all other statistical operations to estimate in an accurate way what can be the isoform expression at cell resolution. What I find really interesting in this pipeline and in the usage of Nextflow is those workflows work in an asynchronous way. So the first part of the workflow can work independently of the second part. And at a point, if we need we have the result file. We can just, they can just connect and then generate the third part of the pipeline. At the very end of the pipeline, we have the capacity to perform a downstream analysis, and that is depending of the user. If the user wants to generate downstream file, he can just give an option, and then we are able to give the set of genes for which we have a differential isoform usage, and also relevant plots which show how those isoforms change in the different cell type of condition. Here is a part where I say that there is a room for improvement using the last feature of Nextflow. Currently, I develop SCALPEL, where SCALPEL can be executed using a Conda environment. Also, the user can provide an apptainer container, which have all the required package for running SCALPEL. And I think that after the 2 days hackathon, I will definitely use the Sekera container platforms to integrate into SCALPEL, which make it very easy to run. So SCALPEL runs as this, and then generates the output file. So we tested SCALPEL to see how much SCALPEL performs compared to the existing tool for quantifying isoform in single-set dataset. So to know at which point SCALPEL performs, we generated a synthetic single-set dataset. So first, we generated a synthetic count gene matrix, and then using an existing tool called Splatter, we generated synthetic population, where we have a population of cluster A, cluster B, which are supposed to express differential isoform. We integrated into the dataset a set of genes, and a very specific subset of those genes show a change in the isoform usage, and other which don't show a change in the isoform usage. So here the challenge will be for SCALPEL to find the right set of genes for which we have a differential expression of the isoform. So based on this synthetic count matrix, a colleague of my lab generated a synthetic FASTQ file. And then as SCALPEL uses those FASTQ files to run, then we can give those FASTQ files to SCALPEL and other published tools, and then see which tool performs the best. So here are the preliminary results that we have, where we see that SCALPEL shows a better precision and recall for identifying genes with differential isoform usage between the cell type. So on the left, you can see that on the gray bar, that we have the number of genes with differential isoform usage that we integrated in the synthetic dataset. And then you can see the other tools which are able to detect only a subset of those genes. And then we have SCALPEL along some other tools which are able to identify the higher number of genes with differential isoform usage. And on the right, you can see the curve of the precision recall curve, where SCALPEL shows a very high sensitivity as well as specificity, then precision in identifying those genes. And importantly is the usage of the isoform information, isoform count matrix, enabled to conserve the cell type specificity. So for people used to do an analysis using the single cell, you are able to generate a UMAP where you see the cell type in a different cell type, as what you can see here. And using the isoform quantification done by SCALPEL, we are able to retain the cell type specificity. But also, which is very important, is we are able to identify new cell state that we were able only to see using the isoform information. So using the isoform information bring a new layer in identifying cell state or cell type, which we cannot see using the classic gene information. As you can see on this figure, we have a new cell type, which is RS6, that we're able to see only using the isoform information generated by SCALPEL. So here we identify in the different cell type what was the number of genes which differential isoform usage. So SCALPEL identify a certain number of genes which change the isoform usage in the different cell type. So here is an example of one of those genes. So we can see that here we have a data set of a spermatogenesis where we go from stem cells in the SC, which differentiate to round cells, and then the ESL, which are mature cell type. And we can see that for this gene, we have a differential usage of the isoform going from the SC cell type, which particularly use the isoform with the long triple UTR. And then when we arrive to the differentiated cells, the ES cells, we have a preferential usage of the isoform with the shorter UTR region. So that is one example of the gene we see with differential isoform change that SCALPEL are able to identify. And if we do an overall analysis, looking to this differentiation path, we are able to see that overall the gene show a switching towards the usage of the short isoform compared to the long isoform in the stem, in the spermatocyte cells. So that is an example of what using a isoform quantification are able to provide in the analysis. So to conclude, SCALPEL is a Nextflow-based pipeline to quantify isoform in the 3' tag single-cell dataset. The SCALPEL quantification is able to retain the cell type specific information. And using SCALPEL, we were able to see that we perform in a better way compared to the actual tool for quantifying isoform in the market. So I would like to thank people of my lab in Barcelona for giving the support for the development of this project. This project is archived. We have already published a first version on bioRxiv, and actually we are pushing for the revision in NatureCom. So for people who are into single cell and want to give it a try, you can come to see me after and ask me some questions, or you can go to the GitHub. Thank you.
Thank you for the great talk. I gotta say, it's very gratifying to hear that the hackathon gave you ideas for further refining the pipeline. I mean, that's perfect. That's exact' why we do these kinds of things. Okay, I have a few questions in Slack. How will the pipeline validate against cases of alternative 5' site or alternative 3' sites beyond just exon inclusion and exon exclusion? And has SCALPEL been validated against long-read sequencing datasets like PacBio?
Yes, the fact that we use a 3' tag, the fact that we use a 3' tag single-cell dataset allows us only to characterize isoform which show a differential terminal region. So is it possible theoretically to adjust SCALPEL to also be able to use 5' tag single-cell dataset and then identify those type of regulation? But for now, we only use a 3' tag single-cell dataset.
That's fair. Oh yes, do the isoform predictions change in accordance with the sequencing technology or chemistry used? For example, 10x 5' versus 3'.
No, there's no difference. Currently, SCALPEL can use 10x and DropSeq. And that depends on the format of the file, mainly. So as long as we are able to have a tool which are able to read broad size of input file, SCALPEL can read different type of single-cell protocol.
Very good. Excellent. Tyler mentions, one challenge I faced with isoform level analyses is the lack of annotations at the isoform level. Does this make cell type annotations challenging?
It's true that you need to provide an isoform annotation for that. So SCALPEL is not able to work without you providing an annotation. So yes, it's a challenge. There is some tools which are able to run without providing an isoform, but yeah, we need to provide an isoform annotation for that.
Saba, yes. Do you have any plans to turn the SCALPEL pipeline into an NF core pipeline?
Yeah, definitely. That's what I saw after the two-day hackathon. I used to think that it would be an easy task, but going with the NF test and all those new features, I think that it's not that easy, and definitely something I will push for it. Mm-hmm. Hopefully, if the paper is accepted in Nature Communication, that will be something to
motivate
make the paper more better. Yeah.
Absolutely. Well, we do have some tutorial resources in development for helping understand some of the more intricate features of NF core pipelines. One more. This is generating a lot of discussion. Harshal is asking, how would the analysis and results look different in disease states like cancer, where you inherently have copy number variations, large structural mutations, and possibly unknown isoform annotations?
In the scientific literature, we know that alternative polyadenylation regulation have been associated in cancer. So, for example, we saw that in some cancer where we have proliferative tumorous cells, we saw that those cells tend to switch to a usage of the short isoform compared to the long isoform. So it's something which have been published before. I haven't tested the SCALPEL on those type of data set. For now, what we did is we tested on the public spermatogenesis dataset, and also we tested on a neuronal dataset where we differentiated neurons from iPS to mature neurons, and then we were able to see that from the iPS cells, from the mature neurons, we see a differential usage of the isoform for the same gene. So there's a lot of new things to discover into this field.
Very interesting. Very interesting. Well, I would definitely encourage you to join the Slack discussion. This is generating a lot of enthusiasm, which is fantastic. Thank you very much.
Thank you.